Let's do this once again. What are we trying to understand? Counter current mechanism. Okay, it's a mechanism by which the kidney is capable of building very high fluid of very high osmotic value. As you go deeper from the cortex into the medulla and further deeper, the osmolarity increases. How is kidney capable of doing that? Is a wonderful question, very interesting question. We are trying to answer that question, and for that we had this example in front of us. So here you can understand there is a a nephron there. You can see the loop of Henle, and to begin with, what did we say? To begin with, what did we say? Let us presume that the fluid is at what osmotic value? 300 milliosmoles per liter everywhere. Okay. And then as the fluid goes here, then on the ascending side, what is the peculiarity? In what way is the ascending side different from the descending side? It has what? It has what? The ascending one. What is it? Peculiarity of the ascending one. It has what? Yes aquaporins or no aquaporins? No aquaporins. What is it? No aquaporins. And second thing is, particularly in the thick segment, okay, it is abundantly rich with the system that can keep on exporting the sodium. Okay, as a result of this, you keep on building a certain concentration difference between the interstitial fluid and what is the content of the ascending limb. And when the pump is working at its best efficiency, it is capable of building how much of difference from across the wall of the tubule? Inside is about 200, outside is about what? 400. No, but since there is no barrier, this equilibrates with the descending. So the fluid that is going in the descending fluid and the interstitium comes to an equilibrium. So everything becomes 400. So you have 400 in the descending, 400 in the interstitial, but 200 in the ascending fluid. We have gone that far. And then eventually, as we know, the fluid flows. Look at this arrow. Okay, and then you have here, so you have even 400, you have 400 because in the descending it was 400, it comes in the ascending, that is 400, it keeps on building the difference. So that becomes eventually from 400 this will become 300, but that will become, and the process goes on repeating till what time? The interstitial fluid it goes to about how much? That's the limit to which 1200, 1300 is the limit to which it can go in the case of human kidney. To what extent it will go in the kidney of that hopping mouse? 10,000. Okay, that's why it is able to. Okay. So now I will take this story forward and I'll ask you. Oh, okay. In this, then we had introduced another character, and that character was called as what? Vasa recta. What did we call it as? Vasa. What is vasa recta? It's a blood vessel. It's a blood vessel. Okay. It's a part of the peritubular capillary network, which is a peculiarity of the cortical nephron or the juxtamedullary nephron. Juxtamedullary nephron. That's the peculiarity. Okay. And then we had seen that the two vessels go parallel, and the flow of the current in them is opposite to one another, and that's what we are looking at it now. Okay. So we have a blown up image of this is the nephron that you can see. So we'll start with 280. Okay. To be more precise, 280, 400, 600, 600, 1000, 1200, 800, 600, 400, 200, 150. Okay. You can clearly appreciate the thick blue line, which indicates what? The absence of what? Absence of aquaporins, and as a result of that, it goes as far as 100. If you go actually beyond, it actually goes to maybe 100 or even less, whatever it is. Now I am going to ask you one question. What may be the nature's rationale in diluting the whole thing? It's diluting, okay? I mean, you went all the way, you concentrated the fluid to what extent? 1200, and then you take up again to the cortex into the distal convoluted tubule, and then you have diluted the fluid to what extent? 150, 100 or whatever, even less than that. I am posing the problem in front of you, and I am asking one of you to answer my question. Please go ahead.
So in the absence of ADH, the urine can excrete out dilute.
You got the answer? Because if you have taken lot of water, then you need to excrete dilute urine, so dilute urine is very important. Okay. So you dilute the urine here. Okay. And if the osmolarity of the blood is less, and if water is a burden on you, and if you need to get rid of the water, just making sure that you don't lose the sodium ions, you have diluted it. So, make sure that you do not absorb any water and throw it out of the body. In what form? Extremely dilute urine. So, that with that, from that point of view, diluting it makes sense, you have to dilute it. Okay. But then what do you do when you need concentrated urine or when you are deprived of water? We will come to that in a few slides downward. Okay. Now, so let us go back to this figure. So, here we have this and coming, so this is vasa recta. This actually, these vessels are very close, they are very close, the two tubes. The one in which the descending tubule and next to that, okay, next to that is the vessel of the venous component of the blood vascular system that is taking the blood supply up. So, there is opposite direction, we have seen that in one of the. Now, let us talk about what happens actually when the blood, so what is this? What is this red one? It is a branch of the peritubular capillary. Good. What is it? It is bringing oxygenated blood. Good. What is it doing? It is sending a branch, it is sending a capillary down deep into the medulla, into that part of the medulla where the osmolarity of the fluid is 1200 meters going there. Now, now this capillary, let us see, now you are here, say the blood is going down, the blood as it enters, what is the osmolarity? 300, okay. Now the blood is going down and it is capillary, and that capillary has many pores. Outside, what is the position? Outside there is more, outside osmolarity is 400, are you with me? It is 500, okay. So, equilibrium will start happening, means what? Outside salts will get in and inside water will go out. So, what is happening to the blood? The blood as it goes down, it keeps on taking the same osmolarity as that of the fluid outside. I am not going to try to keep my identity, no, let me become the same way as the fluid outside, consumes less energy. Okay. So, the salts enter and the water goes down, and then in the vasa recta also, what is the osmolarity of the fluid by the time you reach to the lowermost point of vasa recta? 1200. You are okay so far? Good. Now, and then what have you done in the process? In the process, you have lost your oxygen. That is the purpose of your capillary. You have to give oxygen, and give oxygen to what? Well, you have to give oxygen to this. You have to give oxygen to the loop of Henle. They are the cells, they need oxygen, okay. They need, so you give oxygen to, so all the tissue that is there in the, as you go deeper and deeper into medulla, they need oxygen, you give oxygen and become yourself what? Deoxygenated. Now, as you get deoxygenated, as you start traveling upwards, you do exactly opposite. Means what? As you go, you are in 1200, okay, I am 1200 and there are windows all over, hello, are you with me? And as I go up, osmolarity becomes 1200, 1100, 1000, 900, 800, are you with me? The water gets in, okay, and the salts go out, very simple, okay, and by the time it goes to the top, it is about 325, almost same as blood, okay. So what beauty you have to appreciate here is, whereas on one hand, as long as cells are there, they need oxygen, you have to have the blood supply. But if you just send, now imagine if you and I were to design it, I will say 300, 300, 300, outside is 1200, okay, you will dilute the fluid outside. No, I do not want to dilute the fluid outside, because that pump and that system has worked very hard to make sure that the osmolarity goes as high as 1200, I do not want to dilute it. I am interested in preserving the high osmolarity of the fluid as you go deeper and deeper into the medulla. Have I conveyed to you the paramount importance of vasa recta? Yes or no? Good. Now, second important point is, which I discussed with you yesterday, is we always talk of homeostasis and we always talk of blood always being at 300 milliosmoles. Okay, then how do the endothelial cells, how do those endothelial cells dealing with blood which is having osmolarity of 6, 7, 8, 900, 1100, 1200 milliosmoles, how are those cells coping up? Or even for that matter, how are these cells coping up with the cells of the, I ask a very simple question, there are cells in a loop of Henle, yeah, they are, to what osmolarity they are exposed? 1200, this is 4 times more concentrated than elsewhere. How is this part of the medulla coping up with that? Got the problem? The solution to that problem is as amazing. These particular cells have adapted to them. How they have adapted to them? They have adapted to that by having in their cytoplasm, okay, as many 4 times more solute molecules, so that the intracellular osmolality of these cells. So, if I go here, these cells, the osmolality of these cells is 400 or 600, 900, 800, 1200. So, even those cells are as far as the osmolality of the cytosol of these cells is concerned, they are very different from elsewhere. Okay. So, are you getting the point? So, how these cells have evolved, and that is, this is the kidney. From the kidney you take the, you do the osmolality from, you take samples from 4 places, okay, here, 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 and this is the deepest part in the medulla, okay, and you see the osmolality of the cytosol collected from 4 different cells, and look at the graph yourself. Do you get the answer? I mean, you see, if you were to do this experiment of collecting sample from different cells, different tissues, anywhere, say skin, brain, liver, pancreas, muscle, it is 300, 300, 300, 300, 300. Are you with me? It is only in your body, this is a unique collection of cells which is either in the vasa recta, okay, or in the loop of Henle, where those cells will show you what? Show you what? Osmolality. Osmolality, very osmolality, and that is by, I want you to, here author tells us about what are the different solutes which take the, so notably, can you read further? Polyhydric alcohols and what? Methylamines. So, these are some of the solutes which are highly concentrated in the cells as you go deeper into the medulla, which add to the osmolality, so that the osmolality can be gone as far as, so that these cells are capable of dealing with the highly osmotic fluids which are surrounding. Okay. Now move on. 5 minutes back I spoke about the dilute blood and its importance and its relevance. Okay. Now we are moving, we are going to the other end of the spectrum when the osmolality of blood is rising, because there is loss of water. It's through skin, it's through lungs, it's through feces, it's through dehydration for whatever. So let us see how does your body react when you are being dehydrated. Got the problem? Okay. Now let us see. As a result of that, the blood osmolality, which was, let me go to some precision. The osmolality, you are well hydrated, osmolality of the blood is about 280 milliosmoles. How much it is? 280 milliosmoles. Now, as a result of dehydration, the osmolality of the blood has gone from 280 to 281, 82, 83, 84. Even remember, the rise of about 3 to 4 milliosmoles is good enough to trigger some of the cells in your hypothalamus. So what you have here is just see this, if you take a knife and run it through this plane, so that the human brain is cut into 2 symmetrical parts. Okay, I will call that plane of section as mid sagittal plane. So you cut into part and you look at it, look at the brain, and you will see on the ventral side of the brain, this part is called as the hypothalamus, this part is called as the optic chiasma, this part is called as the mammillary body. Don't bother about it, and this is the pituitary gland. And of the pituitary gland, this is the this human being is facing that way. So this becomes the anterior pituitary, this becomes the posterior pituitary. Are you with me with the basics of anatomy there? What are we doing? We just know in 1 minute we had brief encounter with the hypothalamus hypophyseal system of the human. This is the most important neuroendocrine system of your brain. Part of the brain, hypothalamus is brain, pituitary is the endocrine gland which is controlled by the hypothalamus. Are we good so far? In the hypothalamus, now I will introduce you to very important groups of neurons. Okay. Now I am going to tell them and I want you to remember. Okay. A few things you can't forget. Okay. 2 groups of neurons. There are many groups of neurons. Okay. And I want you to remember 2 groups of neurons. One is called as supraoptic nucleus. What is it called as? It is called as supraoptic neurons because there is, in the hypothalamus there is optic chiasma. Okay, let us do a little bit of anatomy. You have eyes. In the eyes you have retinal ganglion cells. Hello, retinal ganglion cells. The retinal ganglion cells give rise to axons. Those axons, they gather together and they form what you call as the optic nerve. Optic nerve is also the second cranial nerve. Some of the fibers of the second cranial nerve cross right in the middle, right in the cross. When they cross, we call it optic chiasma. What do you call it as? Optic chiasma. Okay, and optic chiasma cut in transverse section is here. So this actually symbolizes what? This symbolizes a large number of fibers from the right retina going to the left side of the brain and from the left going to the right side of the brain. Good so far. Now, sitting on the optic chiasma sitting on slightly on the lateral side of the optic chiasma, there is a group of neurons. Optic chiasma, supraoptic nucleus. Are you getting the rationale why it is named so? What is the name? What name did I tell you just now? Supraoptic nucleus. Okay. Separating the so we have bilateral symmetry, right half, left half. Okay. Right in the middle here somewhere we have a space in the brain which we call as third ventricle. What do you call it as? Third ventricle. Why call it third ventricle? Sometime in future. Third ventricle. Okay. Third ventricle. There is a group of neurons on the left. There is a group of neurons on the right. Paraventricular group. Makes sense at the level of the ventricle. Okay. So, I am talking about these two groups of neurons. What are the two names? Can you please recite for me? Supraoptic and paraventricular. These neurons are very interesting because they secrete two hormones, namely oxytocin and vasopressin. What are the hormones? Two hormones. So, there are some cells, some neurons which secrete a hormone, which secrete a neurohormone, which we call as what? What are the two names that we recited just now? Oxytocin and let us forget about oxytocin for a while. Let us focus on vasopressin. Okay. Now, both the hormones are nanopeptides. Each is made up of 9 amino acid residues. So, how many amino acids are there in vasopressin? 9. How many are there? 9 amino acids. And these axons, these neurons, they give rise to axons. They go down the hypothalamus, and they terminate in the posterior pituitary gland. Where do they terminate? Posterior terminate, and these are neurons. Okay, they are neurons. And in their cytoplasmic machinery, which means endoplasmic reticulum, Golgi, everything, they synthesize a big molecule. That molecule cleaves, and then you have a small molecule which we call as vasopressin, and those molecules are then by trafficking they travel all along the axon and they get into the posterior pituitary gland. And in the posterior pituitary gland, what is the action that is happening? Can somebody look at the diagram and tell me? It's there. The figure on the left. What's happening to the hormone there? It's going into capillaries. Come on, it's so simple. It's going into what? Capillaries. Okay, it's going into the capillaries. Now, that hormone I'll call as a neurohormone. Why do I call it as a neurohormone? It's a neuron. It's neuro coming from a neuron, and hormone is going into the blood. Okay, I can also call the neurons of the supraoptic and paraventricular nucleus as neuroendocrine cells. Good. Neuroendocrine, because they are having a secretion that is going into the blood. So, they fulfill both the conditions. Okay, that they are coming from a neuron, they are going into the blood. So, neuroendocrine. Okay. So, and then the vasopressin gets into the blood, and then it gets into the kidney, and it does its function. So, what function does it do? Let us follow the block diagram. What is point number 1? Water deficit. Very simple. What number 2? Extracellular osmolarity goes up, means from 280 to how much? What I said? 282. I gave you an example. What example did I give you? 285, 284, 285. Very good. So then it stimulates osmoreceptors. Where are the osmoreceptors? Tell me now. Tell me the name. So the neurons of the supraoptic and paraventricular nucleus, they serve as osmosensors. They serve as what? Osmosensors, and they are excited and they give rise to what? Which is also called as antidiuretic hormone. Also called as what? Which is abbreviated as what? ADH. Okay. As a result of this arrow upward, what does it say? What is happening to plasma ADH? Increases. What is happening to now? We are at the level of kidney. Water permeability in the distal tubule collecting duct. What happens to water permeability? Increases, means in the collecting tubule will be absorbed. How it will, we will see. Which means water absorption, which means the osmolarity which was going in the direction from 280 to 81, 82, 83, because now water is retained, it may go back from 84 to 83 to. So, this is our physiological response to the challenge that we are facing. What was the challenge? Dehydration. What was the physiological response? Conserve the water that you have. And there is another equally important response. which is to generate thirst. That generation of thirst will induce a behavior, and the behavior is to look for the water and ingest water, so that your osmolality, which was deviating from 280 in the direction of 280, 283, 84, will be returned back to 80 after on consumption of water. A beautiful image, you will enjoy doing this. Look at this image. We are looking at the same cell under 3 positions. Number 1, on the left, let us see, we have the biological fluid. In the biological fluid, you have a solute, okay, and the author has very nicely coded. Here is some solute and some osmolality. I will call it 280, just call it 280. And if it goes towards from 280, it will goes to 81, 82, 82. You can go in that direction? Can you actually count the number of solutes? You can. And if it becomes dilute, it goes in that direction. Or as if we have given a scale, okay, different osmotic values of the extracellular fluid of the blood. Now, as you are dehydrating, then you are going in that direction. So, the blood is going from 280 to 85, which means very hyperosmotic. So, now you will go to the neuron of the supraoptic nucleus, and the extracellular and the blood in the capillary and the extracellular fluid and osmolality is going from 82, 81, 82, 83, 84. That particular neuron, okay, I will begin with this. Let me go to this point. When it was 280, this cell had the inner okay. Now, in this cell, when this is about 280, I put an electrode outside, I put an electrode inside, and what reading do I get? Minus 60 millivolts. Okay, and when it is minus 60 volts, it is 280. Okay, it's not, there is no challenge here. My neuron keeps on firing with a frequency of, let us see how much it is, say 3 action potentials per second. Are you with me? Just take a number, 3 action potentials per second. Okay. Now, now that was a normal condition, now you move towards dehydration. As a result of that, that is the hyperosmolality. Now, can you see the difference between the size of the neuronal cell body there? What is the difference? The cell is shrinking, okay, because inside osmolality was 280, outside is 285, water will move out, okay. As a result of that, this cell will shrink. The plasma membrane of that cell is equipped with mechanoreceptors. So beautiful, there is no other word, okay. As a result, otherwise, how do you convert the change in the osmolality? How does the cell respond to that? Okay. So, it is a mechanoreceptor. So, there is a mechanical change. Once it shrinks from minus 60, it goes to how much? Minus 55. Minus 55 means it is going towards 0. Hello, it is going to, once it is going towards 0, it is getting depolarized. Once it is getting depolarized, what will happen to the frequency of action potentials per unit time? Increase. Okay. Once then, if that increases, what will happen to the amount of vasopressin being released? More, increase. Let us go in opposite direction. You have taken suddenly 1 liter of water. As a result of that, osmolality has fallen from 282 to 75. As a result of that, now the same cell has gone into what condition now? Tell me, what is the inner reading now? Minus 65, which means the cell is hyperpolarized. Hyperpolarized cell is an inhibited cell. When it is the inhibited cell, number of action potentials are reduced, which means the number of vasopressin that is being released is also reduced. Or I will say that my vasopressin secreting cells are now inhibited, and that is exactly what happens when you drink a lot of water. Okay. But let us go back to the scenario in which
sir,
yes,
mechanoreceptor causing an increase from minus 60 to minus 75.
I won't answer that question now. No, I will send you a paper. Okay. Okay. So, here we have the okay, this is interesting. We are monitoring the plasma vasopressin concentration. Under what conditions? Under the conditions of normal, dehydrate, more dehydrate, okay, more dehydrate here, okay, all right. And then as you do that, what happens to water excretion? Can you see here? When it is dilute urine, you are excreting lot of what? Lot of water, dilute urine, okay. Eventually, the amount of water that is being excreted is less and less because the vasopressin concentration is increasing. We are looking at 2 conditions. We are looking at this brown indicates what? absence of aquaporins. We have done it. What is it? Absence of what? And there are no aquaporins. We have already seen that urine is diluted. That is the function of the diluting segment. It is diluted. Okay. And are there any aquaporins here in the collecting duct? Collecting duct cortical, collecting duct here. Are there any aquaporins? Are there here? Say that again.
Not constituted.
Not constituted. I will tell you what. As a result of no aquaporins, will the water go out? Water won't go out. So that dilute urine will go pass out as it is. Okay. Therefore, we have therefore we have the okay. No, I am sorry, I made a mistake here. There is no aquaporin here, no aquaporin, and then so what is the strength of the urine that is going out? 65, which is very dilute. Okay. Now, here you introduce the aquaporins, and then let us see what happens when the aquaporins are there.